All right, we'll move to the next talk of the session. This is by Diako Ebrahimi from University of Minnesota, and the title of his presentation is Molecular Determinants of HIV Hypermutation.
Good morning, everyone. Human chromosome 22 codes for 7 apobec enzymes. 4 of these enzymes, apobec 3, D, S, F, G, and H, can inhibit HIV through cytosine deamination. And this is how they do it. So most of them are counteracted by the HIV protein VIF, but then if they can escape this process, they can travel with the HIV particle from a producer cell to a target cell where they can mutate cytosine to uracil on the negative strand of the HIV genome during reverse transcription, and this is manifested as a G2A mutation on the positive strand. These viruses can then integrate, and if we sequence them, we see 2 distinct hypermutation signatures I've shown in green and blue. So this signature is primarily within the GG dinucleotide. This is GA, and this is mainly done by apobec 3G. This mutation, the blue mutation, is done by other apobecs, D, F, and H. So if you look at the HIV sequences that you get from the infected patients, you see an interesting pattern. So you see a lot of green signature or a lot of blue signature, but not both at the same time, suggesting that maybe the action of these 2 apobec classes are independent. And if I quantify now this for many HIV sequences from many patients, such that each sequence is represented by the percentage of the green signature versus that of the blue signature, I see this orthogonal pattern, again suggesting that they do not co-mutate the same HIV genome. You see empty space here. Same analysis done on different patients, one at a time, shows again another interesting pattern. Some individuals show primarily the green signature, some show the blue signature, and some have both. And then because the differences are between patients, our first guess was that it's probably due to genetic variation in these apobec enzymes. And we came up with this idea, this hypothesis to explain this. So we hypothesized that maybe a good form of apobec 3G goes with a bad form of other apobec. They are on the same chromosome. And anytime you have a bad form of apobec 3G, it goes with a good form of apobec 3D, F, or H. So 2 good apobecs cannot be on the same chromosome. And this actually gives us 2 alleles, and people with mainly green signature are probably homozygous for allele number 1, in which only apobec 3G is active. People who show the blue signature are probably homozygous for this allele, allele 2, in which only blue works, and then people with this signature are probably heterozygous. To test this potential association between good and bad, and by saying good and bad, I mean good SNPs and bad SNPs in apobec, we performed a genetic linkage analysis of all the SNPs of A3G and all the SNPs of other apobecs enzyme, and blue means no linkage disequilibrium, red means high linkage disequilibrium, and you clearly see that only apobec 3H is highly linked to apobec 3G genetically. And importantly, some of these SNPs that we have identified for apobec 3H have already previously been shown to be important in terms of their role in protein stability, antiviral activity, and localization. So this is how these genetic blocks look like. We have 2 blocks of A3H and A3G. The RDD version of that, A3H, goes with these sets of SNP in A3G, and then the GKE form of A3H goes with these different sets of SNP of A3G. And this is primarily found in sub-Saharan Africa. This is mostly outside Africa. We call this haplotype 2. We call that one haplotype 1. We know already that haplotype 2 produces a potent antiviral protein. This does not. So we have a good apobec here and a bad apobec, blue here. Based on our hypothesis, good goes with bad and bad goes with good. So if someone is homozygous for this genotype, it's A3H, which is doing most of the job, and you see this kind of signature. If someone is homozygous for this genotype, it's A3G, which is winning, and you see the green signature. If someone is heterozygous, they can mutate by both apobec enzyme and you see both signatures. So to test this idea, the first thing we did, we looked at the RNA-seq analysis of cells with known genotypes. And before I show you the results, I'll give you a quick introduction of ApoBec3H because it's a complicated protein. So A3H is the most diverse member of this family. It has 4 splice variants with different number of amino acids shown here. The most important one is this SV200, as I will explain in the next slide. It has this exon called exon 4B, which is an important exon, and it's not included in the other main splice variants of ApoBec3H, 182 and 183. And the genetics of A3H is also complex. 7 haplotypes have been reported for this enzyme. We have identified 12 in the databases, and this is resulted from different combination of these SNPs that you can see there. These haplotypes are not randomly distributed across the globe. Haplotype 1 that you see on top is mainly found outside Africa. Haplotype 2, the second most dominant, dark blue, is found mostly in African populations. So going back to our model, what you can see here is one individual, the RNA-seq profile of one individual with this genotype and one individual with this genotype. And what you can see immediately is that all the A3H exons are the same, expressed at the same level, except this exon 4B, which, as I showed in the previous slide, is responsible for the expression of the SV200 splice variant. So I then went and quantified all the percentage of all the transcripts of ApoBec3H in people who are homozygous for allelic block 1. We call it homozygous HAP1. People who are homozygous for this allele, homozygous HAP2, and heterozygous people in the middle. Again, you can see immediately that the SV200 expression, this purple, is the one that makes the most difference. It is highly expressed in sub-Saharan Africa. It's not expressed as much outside Africa. So now the question is, what is the molecular mechanism? To cut the story short, we did a comprehensive association analysis between all the SNPs and the expression of different splice variants. And we found a deletion right at the upstream of this exon 4B. It is at a potential, polypyrimidine site, which is a potential splicing factor binding site. And what we see here is that if both of these alleles are deleted, sorry, it has to be delta here. If both alleles are deleted, you have the highest expression of this exon, therefore the highest expression of SV200. If none of them are deleted, you have very little expression of this exon and little expression of SV200. If someone is heterozygous for this deletion, it's an intermediate with very high p-value. And one thing to note is that this exon, in fact, the entire exon is part of a line element which has integrated and is pretty ancient. So this brings us to our current working model. In outside Africa, it's A3G, which is winning, and you see the green hypermutation patterns on HIV genome here. Not only these SNPs of A3H are good, but also you have an extra an additional splice variance because of this CTC deletion. So we think that A3H is winning here, and you see the blue signature in HIV sequences hypermutated by this genotype. And then if you have both alleles, you can hypermutate by both. So Chris in our lab became interested in this project and did experiment with this splice variants. The first thing he did, he looked at the blots in a single cycle infectivity assay, and for now focus on the first column. And what you see here is what he observed was that, as expected, a single band inside cells for 183 and a single band inside viral particles. But for SV200 version, he observed a single band in cells but 2 bands in viral particle. And our first guess was that this is probably HIV protease, which cleaves this splice variant specifically. So to test this idea, he used the protease inhibitor drunavir, and then the band went away. So we already knew that ApoBec enzymes interact with the HIV protein VIF. Now we have discovered a new protein in HIV, which interacts specifically with ApoB3H and specifically with this version of ApoB3H, which is prevalent in sub-Saharan Africa. Now the question is, what is the functional consequence of that? So in conclusion, we think that ApoB3G and ApoB3H are the main source of hypermutation main source of HIV hypermutation. And due to this polymorphism and biased geographic distribution, most people use either G or H, but not both at the same time to hypermutate HIV. And then we discovered this CTC deletion, which leads to the expression of an additional splice variant in sub-Saharan Africa, which interacts with HIV protease. So with that, I thank the previous PhD student who worked with me, who did most of the work. He's a postdoc now. People from Harris's lab, particularly Chris, who does most of the experiment, and Michael. And then Jairam and Bruce, who collaborate. Jairam has sent us PBMC from infected patients, and Bruce is helping us with the protease story. Thank you.
Do you know what cleavage does to activity?
Not yet.
Very nice. Is there any kind of clinical outcome data comparing these different genotypes?
Not yet.
If you had to be HIV positive, would you prefer one or the other of these variants?
We don't really have that data. The only thing we know is that in sub-Saharan African populations, HIV sequences reported in Africa, if you analyze the hypermutated patterns, you get something like 50 to 60% of them are GA, they are blue signature. But then outside Africa, the one reported in China, in India, where the haplotype 2 is not prevalent, you rarely see a GA to AA mutation pattern. They are mainly ApoB3G signature. That's another piece of evidence that I didn't have time to show.
First in the back over there.
So this bifurcating mutational signature, couldn't that at least partially be attributable to VIF variation, differential degradation of the ApoBX?
That's a very good question. Our initial analysis of all the subtypes from these patients that we have does not show an association. But if we do more thoroughly...
you can find within a single patient variants of VIF that differentially target the various ApoBX enzymes.
That's possible, but then that doesn't quite explain why in some individuals, despite the fact that they have a lot of quasi-species, we still do not have a single blue signature. They are all green, for example, or vice versa.
None of this shows up in GWAS, and I might have expected, if these really are complementary, that the heterozygotes might have done better. Would you care to comment?
Yeah, the GWAS data is not something associated with hypermutation. It's viral load, other things. I don't think these patterns are associated with that type of clinical information.
Well, I think if you had very effective suppression of viral replication with ApoBX, you would certainly show up as viral load.
Yeah, well, data on the association between hypermutation level and the suppression of HIV is controversial. Some people think that viral load goes down when hypermutation goes up, and some other papers shows the opposite, or they don't show any effect. So we are not sure at the moment.
I'm a believer in hypermutation driving down
You are?
viral load.
Perfect.
Very much so.
In the back.
In one of your early graphs of all the patients with hypermutation, it looked like there was a tail on the end of the A3G signature. In other words, when A3G was really high, there was a significant number that had A3H as well. With respect to Paul's question, is it possible that some of those have VIF variants, and that explains why those have some A3H?
Well, this tail that you see going up, it's actually, you see that in vitro experiment also. So this is where something like 60 to 80% of all the GG position have been hypermutated. There is really nothing else left there. So my guess is that when you get to that level of saturation, all other positions that apoplectic G, for example, can mutate, for example, GA, will appear. And as I said, we do see that in vitro studies also where we only have apoplectic G or only apoplectic E4H.
Right. It didn't look like it was the same effect on the y-axis. There wasn't the same tail, I guess.
Well, it's a little bit tricky. You do see, so in order to be able to present it properly here, what I did was that I only used those individuals for whom I had a single, at least one non-hypermutated sequence. This is why you see less of blue signature, because most of the sequences from the African population, we only have one hypermutated sequence, no normal. So I had to exclude all of that because I had to calculate the percentage based on aligning to a normal sequence within patient. This is why you see less, otherwise you see both tails.
Right, thank you.
One last question.
Is the line element present in chimpanzees?
This line element is present in chimpanzees. In rhesus macaque, some of the tails may be a little bit shorter in some other species. But
so you're saying they all have good
yes.
H's rather than...
Not really, because in chimpanzee and rhesus macaque, we do not have this deletion. It is specific to humans. This is why we have looked at the RNA-seq of monkeys and chimpanzee, and we do not see this SV200 expression.
All right. Thank you.